Good afternoon, ladies and gentlemen. Welcome to this third session of the Scientific Forum, Age Open Science, at this La Salina Palace at Deputación of Salamanca, which is collaborating with us in this event. Longevity is a matter that concerns all of us, and it is such a privilege to hear from the top scientific, from their research about the latest development, and be able to ask them questions about longevity. We would like to welcome the audience who are following these sessions via streaming. Now we are starting calling Maria Holgado, Professor of Pharmacology at the Faculty of Medicine in Salamanca, to come to the stage, please. She's already there. Thanks.
As an investigator, I am delighted to present Thomas Rando from UCLA Broad Stem Research Center. Thomas Rando's research focuses on the biology of muscle stem cells, their aging and the age-related roles in decreased regeneration and the age-related atrophy. He was the first researcher to discover that blood from young mice could reverse the effects of aging in old mice. Today, he will enlighten us on the stem cell aging, functional consequences, and rejuvenation strategies. Thomas Rando, please, the floor is yours.
Muchas gracias, Marina. Buenos días. Thank you for coming to, is this too loud? For coming to Age Open Science. As Marina said, my laboratory studies stem cells and stem cell aging, and I'll tell you today about how we've approached this topic over the last 20 years or so, and our interests in understanding both aging and also rejuvenation and regeneration. So aging is an enigma. We still have a lot of mysteries in terms of why we age and how we age, and what are the things that change as we age that might actually be opportunities for us to slow the aging process and potentially even reverse the aging process. And that's what I'll share with you today, some ideas we have that emerged from our work in stem cell biology and really how tissues repair themselves and what we can learn from that kind of tissue repair and what that leads us to an understanding about the aging process and changes that occur. So clearly, as we age, we have changes that occur that are obvious. We can see the changes that occur in the outside skin and the hair and so forth. But then inside the body, there are changes that are going on in cells and at the molecular level. And really, the last 20 or 30 years has seen a real revolution in terms of our understanding of both cellular and molecular aging, so that we can begin to understand what are the underlying changes that occur that lead to the changes that we see in people as they age. So in my laboratory, we study primarily skeletal muscle, but this could be, the talk that I'll give today could really be given about pretty much any tissue in the body. Now, skeletal muscle is interesting because in the human body, we have 600 and some number of different muscles. And we talk about skeletal muscle as if it's one tissue, and there's obviously similarities, but as you look at the muscles throughout the body, they can be quite different. So the muscles of our limbs are very different than the muscles that control our breathing, that are different than the muscles that control the movements of our eyes, and they come from different parts of the embryo during development. Different diseases affect different muscle groups. So even though I'll talk today very generally about the idea of skeletal muscle, really we could dive down and talk about individual muscles, and they might be somewhat different. One of the characteristics of muscle is that if you injure muscle, it will repair. And there are other tissues in the body, again, that do that very well. You can injure the liver, it will repair. You can injure the skin, it will repair. And yet there are tissues in the body that do not repair well. Parts of the brain will not regenerate. Most of the brain will not regenerate. Most of the heart will not regenerate. But skeletal muscle is one of those tissues that has retained the ability to respond and repair after injury. And I'll just show you an example of that, and this is from studies in the mouse. And as I said, I'm going to talk about regeneration in muscle, but there are many tissues that have very similar kinds of processes that lead to going from a normal tissue to an injured tissue to a repaired tissue. So on the top, what I'm showing is this is just an image under the microscope of skeletal muscle from a mouse. Human muscle looks very much the same. Each one of those polygonal structures is actually a single cell. They're very, very large cells in the body. And they form the units that actually allow our muscles to contract. They contain the proteins that allow your muscles to move and allow you to generate force and strength. In the second panel, we've produced in the muscle an injury, and that injury has destroyed really all of that normal architecture. So what happens then is that the tissue degenerates, you get influx of different inflammatory cells, the debris is cleaned up, and from that debris, within that kind of chaos, there are stem cells, which I'll show you, that then start to repair the tissue. And you start to see in the third panel the beginning of new muscle formation. And then at the bottom panel, you see that muscle having completely reformed. And this actually is a kind of a recapitulation of what happens during development. There's no injury phase, but you do start off with these cells that are very primordial. They begin to divide, they fill up the tissue, then they start to differentiate into these specific kinds of muscle cells, and they grow. So this process of repair is very much like what happens during development, when all tissues are formed from these early stem cells. Now, what I'm showing you here is a higher view of muscle. So again, you see these large polygonal cells. These are the muscle fibers. And right there shown with the arrow is a single stem cell. And they're very rare, actually. They occur infrequently in the muscle, and they sit there as kind of dormant, quiescent cells, only responding to an injury. So that cell could sit there without dividing, without doing anything, for days, weeks, months, and we think in humans even years. And yet in response to an injury, those are the cells that they begin to divide, they fill up the injured area, and they make the new muscle. There are stem cells in most of the tissues of our body that do exactly the same thing. So there's stem cells that sit in the skin, that if a different kind of injury, an injury that's quite severe happens to the skin, they start to divide, they make new skin. There are cells in the liver. If you injure the liver, these stem cells, they begin to divide, they make new liver. And other than, again, those few tissues that don't regenerate at all, these cells can be found through most tissues of the body, and they're really there to repair in response to injury. But we know, one of the things we know, and you'll recognize this gentleman, is that with age, we start to repair our tissues less well. So if you have a skin wound in a young person, in a young individual, that wound will heal very, very quickly, very, very effectively, and essentially without any scar formation. In an older individual, that same kind of wound will heal more slowly, less effectively, and typically with scar formation. So there really are changes that occur with age in this regenerative process that we see pretty much in every tissue in the body. And one of the questions is that ability to repair reflective of other changes that are occurring in the tissue and in the stem cells as we age? And our question really has been, why is that? Why is it that we can't repair our tissues as well when we're older? The first idea is, well, maybe we just run out of stem cells, and that is absolutely not the case. In fact, in some tissues, as we get older, we end up having more stem cells. So it's not a matter of number, it's a matter of function. So we have plenty of stem cells in our tissue, but they don't work as well. So this is, again, an example from skeletal muscle in an injury and repair process. So on the top, I'm showing, again, muscles from mice, and these are young mice. So on the left is uninjured muscle. In the middle is several weeks after an injury that, like I showed, and you see perfect restoration of that architecture. And then after a second injury, a few weeks later, on the right side, again, you see perfect restoration of that tissue architecture. And you can do this over repeatedly. So these stem cells not only make new muscle, but they make new stem cells. So there's this process called self-renewal, where the cells not only give rise to these complex tissues, but they give rise to themselves. So they're always replenishing the stem cell pool. With age, we start to see a change. So uninjured muscle looks really quite normal. But after a first injury, you start to see little areas that are sort of the whitish areas in that image, which are areas of incomplete regeneration and this kind of early scar formation. And then after a second injury, you see an even more dramatic decline in regeneration. It regenerates much more slowly. There's some persistent inflammation, and again, you start to see scarring formation. So again, we knew early on that this is not because of a decline in stem cell number. We know that the number here is sufficient, for example, in a young animal, to repair the muscle perfectly well. So we have over the years begun to study what are the molecular changes, what is the environmental changes that make an old tissue, like muscle, repair less well. And equally interesting, is there really anything we can do about it? Is there any way we can modify either the cells themselves or the environment in which those cells are repairing tissue so that a young tissue can repair as well as an old tissue? And that's the kind of the concept that I'll dive into, which is this idea of rejuvenation. And we see this rejuvenation from the point of view of looking at it from a stem cell perspective. Okay, so this question led us now more than 20 years ago to do this rather odd experiment. And this is an experimental approach called Peribiosis. Now, Peribiosis was first reported in the mid-19th century in laboratories in France where they were connecting different animals to each other. So let me just explain a little bit what this is. So in this procedure, two animals, in this case mice, are surgically connected. And they develop over time, over a few weeks, a single shared circulatory system. So they become sort of like conjoined twins. So the blood of one animal is circulating into the blood system of the other animal and vice versa. So we can connect so-called isochronic pairs, meaning the same age, so a young mouse to a young mouse, or an old mouse to an old mouse. But we can also do these so-called heterochronic pairs, so different ages, a young mouse connected to an old mouse. And our question was, in these experiments, is as we do this, do the circulating factors of the young animal that are now circulating in the tissues of the old animal, do they have an effect on that old tissue? Do they actually have the ability to make that old tissue behave like a younger tissue? And we had evidence from experiments that we'd done that that might be the case. So we set up these pairs, and we let them stay as pairs for several months. And then we start to test the different tissues. We've looked at a variety of different tissues. Again, I'll start with muscle, but what we'll do is we'll injure a muscle of one of these partners, one of these so-called parabionts, one of the partners, and we'll ask, does it regenerate like an old animal? Does it regenerate like a young animal? How has it changed? Oh, so I threw this. So we were not the first to think of this. So this is a painting by Frida Kahlo, which I found, which isn't quite Peribiosis, but she has the concept here. So she has the concept of, again, these are the two Fridas, and she painted this actually following her divorce from Diego Rivera. So there's the traditional Frida on the right, sharing the pain with the more modern Frida on the left, and so she has this idea that there are things that are transferred in the blood from one of her personas to another persona. So this was we were not the first to think of Peribiosis for aging. So okay, so back to our studies. Again, what we'd do is we would set these up, and then we would let them, and then we'd injure tissues or look at tissues and ask how had they changed. And this is just one example from one of our early studies. So if we look at the regeneration of one of the partners in the isochronic young pairs, so this is young paired to young, they regenerate very well. If we look at the partners in one of these old isochronic pairs, so two old animals paired together, on the right you see that whitish area, that's scar formation. So they regenerate just like an old mouse by itself. On the far right you see the regeneration of muscle in the old partner of one of these heterochronic pairs. So this is an old mouse that had been exposed to the circulating factors of a young animal for several months, and it truly regenerates now like a young animal. And so it was really quite a striking finding that you could actually see a restoration of youthful properties just by exposing this old animal to young circulating factors. So we did this initially in muscle and in liver, and we showed that liver also regenerated, an old liver regenerated as well as a young liver. A few years later, we looked at this in terms of brain stem cells. There are areas of the brain that actually do regenerate, that have stem cells, and we found that old animals exposed to this young circulating factors, their stem cells had acquired more youthful characteristics. So again, sort of a rejuvenation of the stem cell function, and also rejuvenation of many different cognitive functions. So there's really a profound effect of this kind of young environment on older on the older tissues. And then over the next decade, many people, and actually Professor Franklin, who you'll hear from next, studied this in terms of remyelination, I think he'll talk about, in the central nervous system. But people have looked at this in a variety of different tissues. So old pancreas, the cells that produce insulin, start to acquire more youthful features in these old animals from peribiosis. Aspects of the heart are rejuvenated. Brain vasculature. So one of the things we know is that with age, there's a reduction in the vasculature in the brain. In these heterochronic parabionts, the old brain vasculature starts to acquire more youthful features. Bone healing, kidney healing, even intervertebral discs in the back start to acquire more youthful features by this peribiosis, and even in the systemic vasculature. So there are tissues in which we see greater effects and less effects, and it sort of depends on how you look at them, but there has been clearly evidence of this idea that there are factors that can really almost regulate the aging process. And one of the things I didn't mention, actually, that I should mention, is that in these heterochronic pairs, the old animal starts to look younger, but the young animal starts to look older. So equally, there are effects from the old circulation in young animals that seem to promote aging. And in fact, they do this quite dramatically. If anything, I would say the effects from the old animal to the young animal are even more dramatic. There really is a promotion of these kind of aging phenotypes from the old to the young. So this led to, obviously, a lot of interest scientifically to try and understand, on the one hand, what's in the blood? You know, what is it that's kind of mediating these effects? And that has led to a lot of studies as well as commercialization, people trying to identify factors that are in young blood that are pro-youth and factors that are in old blood that are kind of pro-aging, trying to understand those, trying to see if we can actually find ways to turn those into therapies, that we could actually figure out from these studies what we could turn into a therapy that you could deliver to people either as a drug or as a protein that might actually restore youthful properties to old cells. So the other side of the equation is trying to understand what does that actually mean to make an old cell young. And this is something that we study in the laboratory still. You will have heard data from other investigators here today, and you'll hear some later, really trying to understand what does it mean for a cell to be reprogrammed from old to young. And are we beginning to see this in terms of using blood to try to manipulate the aging process and see how that changes basically the program of the cell? So this reached a lot of interest also commercially. So this is an image from a situation comedy, a satire actually in the US. It's called Silicon Valley. It's a satire of the tech industry in Silicon Valley. And soon after our papers came out on this, they have an episode in which the tech giant, he's getting regular infusions of blood from this young, very healthy male who he hires to give him blood to make him more youthful and more healthy. So really, this is the question. We clearly are affecting the way these cells behave, either in culture or in the animal itself. We're affecting the way they express genes, but is this truly rejuvenation? And I think this still remains a question in the field because we're clearly modifying these cells, and tissues, and they're clearly acquiring much more youthful characteristics. But are we actually reversing time? I mean, are we actually going back along the direction of aging that they came? We have evidence that that's the case, but I think we still do not have a complete understanding of what that might mean, and that is an active area of investigation. So, initially we used the term rejuvenation and we still use the term rejuvenation quite carefully because it carries a lot of weight, that word. It really does say reversing aging. And it sounds almost science fiction, right? That you could actually take an old person and make them younger. But the process of rejuvenation is so fundamental to biology of life that we take it for granted that this is something that happens and is essential for life. So what I'm just showing here is a fertilized egg. Okay, so this is the cell that will give rise to an entire human being from a single fertilized egg. What the miracle is here is this cell is generated by the fusion of two cells, a sperm and an egg, each of which, no matter how you want to quantify and how you want to characterize the age of a cell, they come from humans that are decades old. So the human is 20, 30, 40 years old, you get a sperm from this person and an egg from that person, you put them together, and you get a cell that by definition has an age of 0. Because otherwise, if that were not the case, with every generation we would be passing on our age to our children, and we don't do that. So the rejuvenation that we were seeing was experimental, but this rejuvenation is absolutely fundamental and is essential to the propagation of life. If it were not for this, we would age ourselves into extinction in several generations, and we don't. So this is an aspect of biology that we just take for granted, that you take two cells of an age, you put them together, and you erase the age. So this is kind of a convergence in our mind of the work that you'll hear about reprogramming, our work on peribiosis, and this really fundamental biology that we know and take for granted is part of normal biology. So we still use the word rejuvenation cautiously, but I think it is at least firmly embedded in our fundamental understanding of many biological processes. Okay, so what I want to end with is just our thinking about how, even if we don't turn this kind of work into, say, drug treatments, are there physiological things that we can do to make our older cells younger? We'd all love to think that's the case. And obviously, peribiosis is not something that we're going to be doing to humans to make them younger, but are there other things we can do? And what I'm going to talk about just very briefly, and we have two studies that are going on this, are the two most fundamental things we talk about in the world of aging, which is diet and exercise. What should I eat and what should I do? And so we've been studying this, again, in laboratory animals to try and get a handle on, are there ways that diet and exercise also contribute to this biology of rejuvenation that we study? So this is an article from the New York Times a few years ago, which I love because I'm a runner. And if you take this to heart, this means you can run and therefore live forever, because the longer you run, the longer you live, which is obviously not true, but it's at least hopeful. So we've been interested in studying the effects of exercise, again, on aging and stem cell function. So for exercise, what we use is these running wheels, and mice actually love to run. They voluntarily do this running. They put this in the cage, and they will run kilometers per night. They will run and run and run. So they really love to run. So this is our exercise intervention. And what we did is we had mice either not running or running, young and old mice, either running or not running, for several weeks, and we basically asked, if we can dive into this biology and we look at their stem cell function, what is the effect of exercise on stem cell function? And again, we were looking at muscle. So I'm not going to tell you too much about the data, but just on that graph on the upper right, it says that with age, so what that is actually, it's a quantification of muscle regeneration. And the first bar shows regeneration in a young animal. The second bar shows regeneration in a young animal that has exercised. And you see really no effect. But the third bar shows regeneration in an old animal. And you see this decline that I mentioned. The fourth bar shows that having exercised for a few weeks, these muscles now regenerate, not as well as a young animal, but much better. So just this three-week bout of exercise can really restore youthful properties to this regenerative response. And we've examined the stem cells from these muscles, and again, they acquire more youthful cellular and molecular features. So just this process of exercise seems to also have some rejuvenating properties. Now, as you all know, if you stop exercise, you quickly lose it, and that happens with the stem cells. The rejuvenation that they get, after a couple of weeks, it goes away. So you have to, the mice have to keep running to maintain this kind of benefit of exercise in terms of their stem cell function. On the bottom, what I'm showing is a graph from studies we did in which we took the blood from an exercised animal and gave that blood to a non-exercised animal. And what we found, like with the peribiosis, that in the blood now are factors that actually make old cells younger. So exercise is not on muscle is not necessarily having a direct effect on the stem cells in muscle because they're in that tissue, but rather it actually produces circulating factors that go to stem cells throughout the body that make old stem cells younger. And we actually followed that up with some studies. So we actually dug down in, and what we found is there's a single key protein called cyclin D1. It's high in these cells that are young. It goes down with age. And what exercise does, it induces cyclin D1 in the old cells, and that turns on this youthful program. So this is a case where we've been able to actually take something as complex as running, and at least in this cell type, in this case, begin to understand at a molecular level how this can actually restore youthful properties to cells. But so I was going to say, we followed this up with some exercise studies looking at brain stem cells, and we saw the same thing. That is, if we take young and old animals with and without exercise, with exercise, we could actually induce a more youthful characteristic of the old stem cells. So the same thing we saw with the Peribiosis. Okay, so let me turn to diet. So in the aging field, again, there's nothing that has been more well studied than diet. And what is known is that if you restrict calories from animals, whether it's flies or worms or mice or even primates, they will live longer and they will be healthier. So there's something about eating as much as we want, which we tend to do. Mice eat as much as they want in the typical laboratory setting. But if we restrict them by, say, 20% or 30%, their lifespans can be extended quite dramatically. And if you want, there's a caloric restriction society that you can join, and you can restrict your own calories for the rest of your life on the hope that that will actually make you healthier, if that makes you happier. But we're interested just kind of, again, in thinking, is this idea of dietary restriction, caloric restriction, is there also something here that is leading to this idea of rejuvenation that can actually impact cells and stem cells throughout the body? Now, we haven't done the experiment where we've taken mice and put them on a calorically restricted diet for a long time, for months or years. Instead, what we've done is we've turned to fasting. And there's increasing evidence that what's called intermittent fasting, fasting periodically, can have the same beneficial effects, or at least similar beneficial effects, of chronic caloric restriction. So we've taken mice and we've fasted them for a day or two, which is a long time for a mouse, and asked, what effect does that fasting, which again you can do, we can all do, have on the biology of their cells in young and old animals. So this is just an experiment in which we've taken the cells from old mice that are either eating as much as they want or fasted. We've put in a probe that has a red fluorescence to it, and then we've transplanted those cells into another mouse. And what you see on the left is the old cells from the animals that ate as much as they wanted. They produced a little bit of muscle. So you're looking at a whole muscle here. There are a few muscle fibers that are formed. But if we first fasted those mice, or another group of mice, took those cells and transplanted them, they were much, much more robust in this transplantation assay. They could make muscle much better. And again, we dove down into the molecular analysis, and these cells also have a restoration of a kind of youthful features. And again, these are some details I won't go into, but as many of you know, when you fast, in your liver you produce ketones and you become so-called ketotic. These ketone bodies travel through the blood, they enter cells, and they actually they're used for energy. Since you're not eating, these ketone bodies are used for energy to produce ATP, the molecule that generates energy for your cell. But what we found, which is interesting, is that the cell has found a way to use this same metabolite. This metabolite that is used to generate energy, it actually uses it to turn on a youthful program. It turns on a resilience program. And so this one molecule, BHB, which is beta-hydroxybutyrate, you can actually buy this in a health food store, you can take it. It's used for energy and fasting, but it also has a completely separate effect in terms of inducing cellular resilience. So this has been, again, a very key finding for us because it links this biology of aging with the biology of metabolism. And one of the reasons we think that fasting is so beneficial to you. So again, let me just end here. So this is the fountain of youth, and are we really looking at ways to restore youthfulness to old cells, tissues, and organisms? And I think the talks that you'll hear today are really converging on this idea that this is not only possible, but we're beginning to understand this at a very molecular level, and that this does have the potential to translate, we think, into therapies for age-related diseases and aging as we move forward in the field. So these are the people who do all the work. I get to report all their great progress, and I'll stop there, and thank you very much for your attention.
Thank you very much, Thomas. Now we are welcome Arantxa Tabernero, Professor of Biochemistry and Molecular Biology. Her laboratory at the Neuroscience Institute of Castilla y León studies brain tumors and the role of neurostem cells on these tumors. Arantxa will introduce our second speaker of this stage. So welcome, Arantxa.
It's a pleasure for me to be here today and to have the opportunity to listen to these incredible speakers. In fact, I have the fortune to introduce one of them, Robin Franklin from the WELLCOME MRC Cambridge Stem Cell Institute. He's at the forefront of studying the mechanisms of remyelination, the process of myelin regeneration. His laboratory has provided insights into how adult stem cells differentiate into oligodendrocytes, the cells that form myelin in the central nervous system, and other types of glial cells. And they have also studied how this process is altered by aging. Robin Franklin has many honors. I'm just going to mention just a few of them. He's a fellow of the Academy of Medical Science and a fellow of the Royal Society. He won the 2017 Baranczyk Prize for Research Innovation and the 2021 King Faisal Prize for Medicine. So without further delay, I give the floor to Robin Franklin to talk about the regenerative biology of adult central nervous system progenitors, reversing the ravages of time. Thank you very much, Robin.
Thank you, Rancho. Muchas gracias. That very kind introduction. So I studied tissue regeneration, which has always struck me as one of the many, if not the most remarkable thing in biology. That we form multicellular organisms is of itself remarkable. But then once formed, they can regenerate themselves when damaged. I think it's, to the best of my knowledge, unique in the known universe. So as a biologist, this has always struck me as something which you cannot help but to be fascinated by. Now, as Tom pointed out, and actually Tom's talk was a very useful introduction to a lot of what I have to say, so thank you for that. As Tom pointed out, different tissues in the body have different intrinsic capacities to regenerate. And the brain, of course, is notoriously thought of as being an organ that notoriously has very poor regenerative capacity. And I couldn't come to this wonderful city, this wonderful country, without acknowledging one of your greatest scientists, perhaps one of the greatest scientists of all time. And in other audiences, I have no hesitation in pronouncing his name, but I feel slightly intimidated in front of this audience in pronouncing it. But I'm going to have a go. So don't wince if it's too far off. Santiago Ramon y Cajal. Okay, there's some thumbs up, so I don't feel too bad about that. So Cajal, he studied many things that we as neuroscientists still study. And he also studied how the nervous system is degenerated and regenerates. And this is a quote from his famous book, which I translated into English, but of course it would originally be written in Spanish. Now, this is sort of a flip image or flip notion of the quote from Darwin this morning that Juan Carlos told you, which was translated into Spanish from the English. So what Cajal said was that once development has ended, the founts of growth and regeneration dried up irrevocably, everything may die, nothing may regenerate. So this is a rather bleak view of the ability of the brain and spinal cord, the central nervous system, to regenerate. Now, at the risk of committing a heresy, Cajal was only partially correct. So this statement is undoubtedly true of one of the cellular constituents of the nervous system, the neuron or nerve cell. And it is true that neurons, once lost, aren't spontaneously regenerated, or extremely rarely in very specific parts of the brain. And it is also true that if you damage the process of a neuron, the axon, it doesn't spontaneously regenerate, at least not in the central nervous system. This phenomenon does occur in the peripheral nervous system. But there are, of course, other equally important cell types within the central nervous system, which are listed here. The oligodendrocyte, the astrocyte, and then the cell of the innate immune response, the microglial cell, which takes up residence in the central nervous system. Of course, it's not a bona fide brain cell. And Cajal's statement actually is unequivocally not true of the myelinating cell, the myelin sheath-forming cell of the central nervous system, the oligodendrocyte. And it's the regenerative biology of this cell that I have studied for a number of decades. So at the top, you see a very simple representation of the brain with 3 neurons with their processes heading off to the right, and they are covered in a myelin sheath, which is made by the oligodendrocyte. And there are a number of diseases where the primary target is the oligodendrocyte, not the neuron. And the best example of that is the human autoimmune disease, multiple sclerosis, a chronic demyelinating disease. Now, when you lose oligodendrocytes in the central nervous system, in profound contrast to the loss of neurons, the default response is a regenerative process which involves the generation of new oligodendrocytes. And those new oligodendrocytes make new myelin sheaths. So this is a true regenerative process. The architecture of the tissue is restored to normality, and function is restored as well. So where do these new oligodendrocytes come from? Well, the oligodendrocyte, like the neuron, is a post-mitotic cell. So the oligodendrocyte doesn't have the capacity to beget new oligodendrocytes. Instead, they come from a very widespread population of cells that are present throughout the whole of the central nervous system. And they are, if you like, the adult stem cell of the central nervous system. They far outnumber the stem cells that occur in the so-called neurogenic niches of the hippocampus and the subventricular zone. But they tend to be called progenitor cells, and you know, whether we call them adult stem cells or progenitor cells, we don't have time today to get into that debate. They just happen for historical reasons to be called progenitor cells, and more specifically, they're called oligodendrocyte progenitor cells, or OPCs. Now, this is an image from a mouse, a reporter mouse that we generated, where the red and the green cells are the progenitors. So this is an adult mouse. The white structures are blood vessels. And the eagle-eyed amongst you will be wondering whether the brain is indeed completely symmetrical. That isn't the case. We've just imaged one side, copied it, flipped it over to give you a notion of the entire brain. So what this image is supposed to indicate to you, and I think does very powerfully, is that this is a very abundant population of adult stem cells or progenitor cells. And in fact, they constitute probably up to 6% of all the cells in the mammalian brain. And if you were able to generate a similar reporter notion for humans, the human brain would look exactly the same. So these are very abundant progenitor cells. And not dissimilar to the stem cells that Tom talked about, for the most part, they just hang around not doing very much. Or if they're doing something, we're not entirely clear what that is at present. So how do they generate new oligodendrocytes? Well, this is a sequence which describes the processes involved in making new oligodendrocytes, and it is very typical of how adult tissues regenerate. So in some ways, paradoxically, this regenerative process in the brain is almost a paradigmatic example of tissue regeneration. So in response to the loss of oligodendrocytes, there is activation of the immune cells, the microglia and the astrocytes, which changes the environment of the tissue very profoundly. And that involves the secretion of molecules that activate these stem cells and induce their proliferation and migration. So following activation, they then divide and migrate like mad, and they fill up the area of oligodendrocyte loss. And then, for the most part, they will differentiate into new oligodendrocytes, and some of them retain the properties of a progenitor cell to replenish or self-renew the progenitor cell population. So that's the simple steps which involve the generation of new oligodendrocytes from progenitor cell. Now, MS is a chronic demyelinating disease. This is a disease commonly diagnosed when individuals in their early 20s, but their life expectancy is only reduced by 5 or 6 years. So this is a disease that many people will have for many decades. And as you progress through life with this disease, the efficiency of this regenerative process declines. It becomes progressively less efficient to the point where, to all intents and purposes, it fails. And this leaves the nerve fibers without a myelin sheath. Now, the nerve fibers depend for their survival on a healthy myelin sheath. So if you don't put the myelin sheath back, those nerve fibers will undergo an irreversible degeneration. And this, for those of you who are familiar with multiple sclerosis, is why a point is reached where the disease stops being a relapsing, remitting phase, where there's loss of oligodendrocytes and regeneration of oligodendrocytes, to a disease that is progressive, and it is progressive because of the continual loss of nerve fibers as a result of the failure of remyelination. So over the years, there has been much head-scratching about why this process should become less efficient with disease progression. A number of years ago, we put forward the totally radical view that perhaps it had something to do with aging. So why does this process decline with disease progression? And we've put forward the notion, which now the field is coming around to thinking is probably correct, that this has to do with aging. Because as Tom has already introduced, probably the single most important parameter of regeneration in any tissue and any organism is aging, which is why we have become fixated in the last few years on the effects of aging on this particular regenerative process. So what we argued is that with time, the function of the progenitor cell decreases. As Tom said, the population of adult stem cells doesn't decrease with age, and that is true of this population of progenitor cells. The numbers of these progenitor cells remains the same. So provision of progenitor cells is not rate limiting. So there must be a change in its function, which is rendering it less efficient at going through these various steps in the regenerative process and making it harder for them to become oligodendrocytes. So a number of years ago, we spent a lot of time developing protocols for growing progenitor cells from aged laboratory rodents. And this has been a real bottleneck in the field. It's very easy to grow, well, relatively easy to grow these cells from neonates in a developmental context. Quite difficult to grow them from animals that are just sexually mature at the age of 1 month. Extremely difficult to grow them from aged animals. So that's why we invest a lot of time developing protocols which enabled us to do that. And of course, once you can grow these cells and study them in a tissue culture dish, all sorts of possibilities open up. So one of the first things we discovered, which was no great surprise, as time passes, the progenitor cell acquires the classical hallmarks of aging. And this would be true of pretty well all populations of adult stem cells involved in tissue regeneration. So here's just one example. There's an increase in DNA damage within the aging progenitor cell as a result of failure of DNA repair mechanisms. So to summarize a very detailed analysis of the aging progenitor cell, in a young adult progenitor cell, these cells do what adult stem cells and progenitor cells do with great efficiency. They divide, and they are able in a timely way to differentiate. As you get older, their stem cell potential declines as the number of aging hallmarks increases, to the point that in the aged central nervous system, these cells enter cell cycle very infrequently and with some difficulty, and they certainly struggle to undergo differentiation and become new oligodendrocytes. So, speaking to a theme that Tom has already introduced, what can we do to reverse this? Can we send this backwards? In other words, can we restore normal function to an aged progenitor cell? So the first thing we did was, we've addressed both diet and exercise. I'll just say, first off, without reference to any slide, we can't find much evidence that exercise reverses the function of the aging progenitor cell. But it turns out that calorie restriction, restricting caloric intake by alternate day fasting, has a really profound effect on the remyelination in aged animals. So just to go through this very quickly, we take animals that are already 12 months of age, and over the next 6 months, we feed one group ad libitum, and they become fat and obese, as you might expect. And the other group we calorie restrict by alternate day fasting. And then when they're at the ripe old age of 18 months, which for a rat is getting on, we induce experimental demyelination, so we kill oligodendrocytes, and then we follow the regenerative process. And the ad libitum fed animals, the remyelination is extremely poor, just as we have shown on many occasions in the past and just as one would predict. But in the calorie-restricted animals, the regeneration of myelin is as if these were 1-month-old animals. And this, frankly, was, at the time, the most spectacular result we had ever achieved in the context of this regenerative process. This was night and day, black and white. They are non-overlapping data sets, which is unusual in biological experiments. So calorie restriction works perfectly at reversing the effects of aging on this regenerative process in the brain of remyelination. So we went to our clinical colleagues and said, well, we've solved this for you. You need to tell your chronically ill MS patients to eat less and give up feeding. And of course, their response was one of some ridicule that they would suggest such a thing. And their response was, well, can you do this with drugs? So the ball was back in our court. Can you phenocopy, can you repeat the results of calorie restriction, but using a drug? Now, there are a number of so-called calorie restriction mimetics. And one of those is a drug that is increasingly widely prescribed, the drug metformin, which is increasingly widely prescribed because it's the drug for treating type 2 diabetes, which is increasingly occurring as the population gets increasingly obese. So what we were able to show, this was quite remarkable to us, that we could completely phenocopy the effects of calorestriction by feeding animals ad libitum, but giving them metformin. And so metformin has exactly the same effects. You can reverse the effects of aging on this regenerative process in the brain with metformin. And what it seems to do is to recalibrate the age state of the progenitor cells so that they proliferate more effectively, they differentiate more effectively, and importantly, they become responsive to a whole slew of drugs that are being touted as regenerative medicines for MS that have the function of inducing differentiation, which don't work in aged progenitor cells, but will work in aged progenitor cells that have been pretreated with metformin. Okay, so let me just move on to the next phase of my talk. And this was work that I did with a physicist colleague of mine, Kevin Shalhoub. It was actually the work of a PhD student we shared, Mikey Siegel. So what we asked here was the fundamental question: why do the properties of this progenitor cell change with age? What's going on? And I've always been of the belief that the best experiments come from answering the most straightforward of questions. So the first question we asked was, well, is aging intrinsic to the progenitor cell, or is it extrinsic to the progenitor cell? So is there some internal drive of aging that occurs within this population, or are they simply reflecting the age of their environment? So you can do a very simple experiment. You can take aged progenitor cells that don't proliferate and differentiate, and you can put them into the developing central nervous system, an environment where we know progenitors divide and migrate and differentiate because that's what they have to do in development. So if aging was intrinsic, the aged stem cells would continue to be in stasis. They'd continue to not proliferate and not differentiate. But in fact, the result was the complete opposite of that. When you put the aged progenitor cells into the developing central nervous system, they now, sorry, I'm getting ahead of myself. Here's the data. They now proliferate and differentiate in exactly the same efficiency, exactly the same as the endogenous neonatal progenitor cells, the progenitor cells of youth. So that tells you actually that the aging of these progenitor cells is not intrinsic. Instead, it has something to do with their environment. There must be something to do with the aging environment, or niche to use the fashionable term, that is inducing a change in the properties of the aging progenitor cell. So what is that change that is occurring in the aging brain? Well, here we just struck lucky. So we had another physicist colleague, a friend of ours, who had a machine called an atomic force microscope. And for no better reason that he had an atomic force microscope, we decided, which measures the stiffness of tissue, it measures the mechanical properties of tissue. We thought it would be interesting to look at the mechanical properties of the aging brain. Just as well we did, because it turned out that in fact the brain increases in stiffness quite dramatically. This is all work done in rodents, because you have to get fresh brain on this particular machine. So what I'm going to describe to you now is rodents. We've no reason to believe this wouldn't be true across all mammals. So just to put it in some sort of context, the young adult brain of a mouse has a stiffness akin to a ripe avocado, but the aged mouse brain has a stiffness that is akin to an unripe avocado. And that is something very tangible and something you can get your head around, and something that is really quite surprising. That's quite a dramatic change in the stiffness of the brain. So does the stiffness per se induce the change in phenotype of the adult progenitor cell population? So as a biologist, I was highly skeptical that that hypothesis would prove true. But my physicist colleagues, not hung up with years and years of rather blinkered view about how cells work, said, well, let's just have a look at this and see. So what we did was, again, a very simple experiment. We took a decellularized brain from a young animal and a decellularized brain from an old animal. So you form a matrix, a platform of the non-cellular constituents of the brain. And you put the aged progenitor cells on the young brain and the young progenitor cells on the old brain. And within a matter of days, the progenitor cells assume the properties of the age of the brain, the substrate, rather than their age of origin. Okay, so that struck me as pretty straightforward ECM-integrin interaction. That would be the biologist's immediate response to such an observation. But my physicist friends said, well, let's go a step further. Now let's see whether this is really actually specifically related to the tissue mechanics. So what Kevin, my physicist colleague, was able to do was generate biochemically inert hydrogels that attune to the stiffness of a young or an old brain. And in fact, you get exactly the same phenomenon. So you put an aged brain on a substrate that is like this, the young brain, and a young progenitor cell on a substrate that is tuned to the stiffness of the old brain, and hey presto, they assume the age of the brain rather than their origin. So a young progenitor cell on a stiff substrate falls out of the cell cycle and won't differentiate, and an aged progenitor cell, which in normal circumstances is useless at engaging remyelination, starts to proliferate and divide. Okay, so that's great. So we now asked ourselves, what can we do to soften the aging brain? And after a while, we thought that was a stupid thing to do, because actually there is some reason why you stiffen, and it has to do with stabilizing synapses. So if you start softening the brain and destabilizing synapses, you could actually wipe all your memories, which probably wouldn't be a very attractive proposition for most people. So Mikey, the PhD student, actually came up with the bright idea. Instead of softening the brain, why don't we figure out how it is that progenitor cells know that they're on a soft or a stiff brain? So this must be by the presence of so-called mechanosensing receptors, which are increasingly studied and for which the Nobel Prize is in fact awarded last year. And again, to cut a fairly long story short, so I'll just skip over that one, we discovered that piezo-1, a mechanosensing receptor, is the molecule that enables a progenitor cell to know whether it's on a soft or stiff substrate. So if you take out piezo-1 from an aged progenitor cell and put it on a stiff substrate, it doesn't know it's on a stiff substrate, so it becomes a young progenitor cell. It starts to divide, and it starts to proliferate. Same thing, excuse me, it starts to differentiate. So this is data in vitro in a tissue culture dish. But then what we then did was to develop a way where we could actually take out piezo-1 specifically in the progenitor cells via a systemically delivered AAV and see whether we could change the efficiency of the regenerative process. So that's the data that is shown here. So when we use a live animal and induce demyelination in vivo, and then use a viral vector to specifically take out piezo-1 exclusively in the progenitor cells no longer they know they're in an aged brain, they proliferate and they differentiate, and so remyelination is made more efficient. So actually, if there were antagonists or drugs that antagonize piezo-1, they would be quite useful candidates as a potential regenerative medicine in a demyelinating disease, in a chronic demyelinating disease. Okay, I'm just going to finish very quickly. I don't know how much time I have left, but do I have time for one more story? Okay, I'll whip through it fairly quickly. So this is a concept that has already been introduced during the course of this meeting. So the phenomenon of reprogramming, taking a somatic cell back to pluripotency, is the ultimate example of rejuvenation. So we were thinking in the lab that maybe you could, instead of taking a cell all the way back to pluripotency, you could take an aged cell back to a youthful state by just exposing reprogramming factors to an aged cell for a period of time. Now, while we were thinking about this, Juan Carlos actually published this in a seminal paper in Cell, which you spoke about this morning, which showed as a proof of principle that this phenomenon was something that was really worth pursuing. So we were immensely excited about this. So could you use the reprogramming factors, the famous Yamanaka quartet of transcription factors, to take an aged progenitor cell back to a young progenitor cell? And the answer to that question was yes, indeed, you could do that. And this has now been shown for a number of tissues. So this is a very established phenomenon. So partial reprogramming will change the age state of a progenitor or stem cell. So given that this was actually not new news anymore, we then asked the question whether actually needed all four of these reprogramming transcription factors. I mean, David Sinclair, as it happens, has also asked this question, come up with a different result. So to cut a long story short, and without going through all of the logic about how we came to this conclusion, what we discovered was that we only needed one of the transcription factors, and that was MYC. Of course, well known as an oncogene, but all oncogenes have daytime jobs, and they're not there as oncogenes in the first instance. It's only in a mutated state. We discovered that MYC alone was able to change the age state of the progenitor cell. So if you take an aged progenitor cell and overexpress MYC, it's enter cell cycle and it will differentiate. Then conversely, if you take a young progenitor cell and inhibit MYC, and we've done this genetically and pharmacologically, then the young progenitor cell will drop out of cell cycle and struggle to differentiate. So that was really quite remarkable to us. So it seemed, oh, and just to make a final point relevant to in vivo regeneration, we used our same AAV CRISPR gene editing strategy to take MYC out of progenitor cells, and we showed that there was an enhancement of regeneration in aged animals. Sorry, to overexpress MYC in aged progenitor cells, and there was an enhancement of remyelination in aged animal cells. So this is quite remarkable. This is a brain regenerative process that is failing as a result of age, and we change a single transcription factor, and we can make it work more efficiently. So that led us to this model that actually MYC, and we don't know what's upstream of MYC, and we don't know what's downstream of MYC yet. Those are experiments for the future. But this single transcription factor is sort of determining the age state of the progenitor cell, to the point where actually now we don't even think in terms of an age state of progenitor cell, whether it's young or old. It's all about, and I think you made this point, Tom, it's all about function. You know, do these cells work optimally or not? So I haven't described to you very much that is translational or clinical, because we haven't got there yet. But I think this work and work of many laboratories will be the basis for translational developments over the next few years. I'm certain of that. Timescales, I can't tell you, and that's always an invidious question to try and answer. But that's just to give you a sense of the work that we've done in the context of a brain regenerative process. And I'm sure Kahlo would have been delighted to hear about this and discover that his rather bleak notion about Peribiosis model, fasting metformin, butyrate actually does exactly the same thing as fasting, piezo-1 deletion, and partial reprogramming. And I should just thank the members of the lab and various collaborators and funders.
For Thomas Rando, is there anything people can do to help rejuvenate our cells?
So I guess that was the basis of my talk. So I would say there are, and people ask me this all the time, is, you know, what should I do? What should I take? And although I did talk about diet and exercise, and as Professor Franklin said, sometimes exercise is less robust, I think, you know, from the wealth of evidence, the simplest thing you can do is maintain a lean diet. That is, maintain low-calorie intake, and I think there are a few things that we have discovered across, you know, the wide spectrum of biology that have so profound effects on health span, meaning prevention of disease, and lifespan than just reducing calories. I mean, I think it's quite artificial. In fact, in the laboratory, we feed mice continuously. They eat continuously, and that is not how animals evolved in the wild. They were not evolved to eat continuously, nor were we. We built societies so we can eat continuously. We put laboratory animals in cages so they can eat continuously, and we have definitely changed the metabolism. So I would say, you know, keep a low-calorie diet. I fast 1 day a week. I have no idea if it does any good, but I do it out of just a belief that it probably will. So that would be my first suggestion.
Mm-hmm. Thank you. Second question is for Robin. If we could stop the deterioration, would we be all human beings with younger bodies?
That's a very interesting question, and a slightly philosophical question. You know, in some ways, I think, you know, it's not helpful to think in terms of young and old. It's whether we work or not. You know, it's all about function. You know, whether we retain function. So I think actually these interventions that we've all been discussing and believe and hope will become interventions of the future, what I sort of envisage is that we won't lose the wisdom of aging. You know, so all of those life experiences will still be retained, but we'll be living those lives in bodies where we sustain as near to optimal function for as long as possible.
Mm-hmm. Thank you. Thomas, the next one is for you. Why do we need to find therapies that have a positive impact on the aging process?
So I would say why we need to do it. I think that the imperative, I think, societal imperative is not to live longer, which many people are interested in. I think the societal imperative is to find treatments that target the aging process so that we can delay and prevent the onset of age-related diseases. Because it's unequivocal that the burden on families and individuals, the economy, occurs because of this burden of late life diseases that we get that is only a function of age. And if there's a real goal in developing these therapies, it's not to extend lifespan, it's to have people live, which is what Robin just said, as healthy as possible, as long as possible. And I think that is the real promise, and that is the real imperative of developing these therapies.
Mm-hmm. Thank you. Last question for Robin. What is the difference between stopping the deterioration of our body and rejuvenating it?
Another interesting question. Well, if I could rephrase the question, it's really about two different phenomena: slowing the aging process and reversing the aging process. I think we already have, as Thomas said, a number of ways that we can effectively change the aging process, and all the lifestyle advice for which there is now increasing compelling scientific evidence will contribute to the changing of the nature of the aging process. Rejuvenation is really sort of reversing the aging process. Well, without getting into the philosophical debates about the semantics of that, and of course remember time is the fourth dimension and it's a physical phenomenon that we can't do anything about. The rejuvenation interventions, I think if we think of them again, to repeat myself, if we think of them in terms of restoring or maintaining function or restoring function to cells that are losing function, then that's a difference from preventing deterioration. So changing the nature of the aging process, and which we can do, and in the future perhaps reversing the aging process, or restoring function, which is probably a more healthy way of looking at it. Yeah.
Thank you. So now we have come to the end, so thank you so much. And this is the first day of Age Open Science. We look forward to seeing you tomorrow again at 10 a.m. in the morning at Patio Chico to continue to talking and understanding the keys of longevity from our expert. We would like to thank you again, our guests, for the participation. And it has been a pleasure to have you here, and now we may have lunch that I bet we are all starving. Thank you so much.
Thank you.
Thank you.
Thank you.